To understand how infection with high-risk HPV can lead to cervical cancer and to explore the different types of HPV tests, let's first consider the structure of the virus. HPV consists of a capsid surrounding a circular viral genome. The genome contains genes for capsid proteins L1 and L2, as well as genes that are involved in replication of the virus, such as E2, E6, and E7. When HPV infects a cell, it sheds its capsid, and the circular HPV genome becomes linear and enters the cell nucleus. As the HPV DNA becomes linear, the L1 gene can be deregulated or deleted. Before an HPV infection can cause cervical cancer, HPV DNA is integrated with the DNA of the infected cell, and the E2 gene is suppressed or deleted, which deregulates the E6 and E7 oncogenes. Many viral genes are deleted following integration, but E6 and E7 must be consistently expressed in order for the infection to progress to cancer. The viral DNA is transcribed to produce messenger RNA, or mRNA, which is then translated to produce viral proteins. The E6 and E7 mRNA creates proteins that interfere with genes responsible for normal cell reproduction. Because the E6 and E7 genes play an important role in dysregulation of cell growth, they are important in identifying HPV infections that will most likely lead to cervical disease. Other genes in the HPV virus, such as L1 and E2, can be lost. If these genes are lost in the beginning of the replication process, they will not be included in the replicated viral DNA and can ultimately lead to a false negative screening result if an HPV test that detects L1 DNA is used. The Aptima HPV assay detects E6, E7 mRNA, which identifies present and active HPV infections.